Hello everyone, you're welcome to today's class. My name is Adebanke. I'll be taking you through top NEET Biology Past Questions. So let's get started. Question 1. Given below are two statements. One is labeled as assertion and the other is labeled as reason. So what do you mean by assertion? Just like a fact, while reason supports assertion, like a supportive statement. So assertion, the primary function of Golgi apparatus is to package the materials made by the endoplasmic reticulum and deliver it to intracellular targets and outside the cell. The assertion is talking about the function of Golgi apparatus, which is to package the materials that are produced by the endoplasmic reticulum and deliver it to intracellular targets and outside the cell. So let's take a look at the reason. Does it support the fact? So reason, vesicles containing materials made by the endoplasmic reticulum fuse with the cis phase of the Golgi apparatus and they are modified and released from the trans phase of the Golgi apparatus. So what this reason is saying is supporting the assertion statement. So both the assertion and the reason, they are correct. So the reason is supporting. So let's take a look at the option. Option A is true, but R is false. Option B, A is false, but R, the reason, is true. Option C, both assertion A and R reason are true, and R is the correct explanation of A. Option D, both A and R are true, and R is not the correct explanation of A. When you look at the assertion and the reason statements, they are both true, and R is supporting the fact. So option C, both A and R are true, and R is the correct explanation of A. Option C is the correct answer. Question 2. A specialized membranous structure in a prokaryotic cell which helps in cell wall formation, DNA replication, and respiration is option A, crystalline, option B, endoplasmic reticulum, option C, mesosome, option D, chromatophores. So when you look at mesosome, it is a specialized membranous structure that is present in prokaryotic cell. It helps in cell wall formation, also in DNA replication and respiration. The correct answer is mesosome, but crystalline is present in the mitochondria, endoplasmic reticulum. Both option A and option B are present in eukaryotes, while option D is only present in photosynthetic bacteria. So the correct answer is mesosome, option C. Question 3. Identify the step in tricarboxylic acid cycle. What is tricarboxylic acid cycle? Is also known as citric acid cycle or Krebs cycle, which does not involve oxidation of substrate. So what is oxidation? That's loss of electron and gain of oxygen. So the tricarboxylic acid is involved in different processes. That's from citrate to isocitrate, associated to alpha-ketoglutarate, alpha-ketoglutarate to cysteine CoA, and the steps goes on and on in the presence of different enzymes to catalyze the reaction. So looking at question 3, so identify the step in TCA cycle which does not involve oxidation of substrate. So the only step in TCA cycle that does not involve oxidation of substrate is the conversion of succinyl-CoA to succinic acid. Option C is the correct answer. Question 4. How many times decarboxylation occurs during each TCA cycle? So we are still talking about the Krebs cycle. The decarboxylation occurs only in step 1 and step 2. That's when acetylcitrate is converted to alpha-ketoglutarate and alpha-ketoglutarate is converted to succinyl-CoA. So this process involves decarboxylation, that's removal of CO2. So the correct answer is twice. So decarboxylation occurs during TCA cycle twice. Option D is the correct answer. Question 5. Fatty acid relates to respiratory pathway through, when we talk about fatty acid pathway, it's also in beta oxidation, and it generates a molecule known as acetyl-CoA, and this acetyl-CoA is involved in other metabolic pathways such as electron transport chain. So, and it's also involved in oxidative phosphorylation and TCA cycle. So, fatty acid relates to respiratory pathway through A. acetyl-CoA, option B. ketoglutaric acid, option D. dihydroacetone phosphate, option D. pyruvic acid, pyruvate. So, the correct answer is acetyl-CoA. Fatty acid relates to the respiratory pathway through acetyl-CoA, option A. Question 6. Which of the following combinations is required for chemiosmosis? So, what is chemiosmosis? Has to do with generation of adenine triphosphate, that's generation of ATP, generation of energy. So, this chemiosmosis needs components such as membrane, and it needs proton pump, proton gradient, and ATP synthase are required in ATP generation. So looking at the question, which of the following combinations is required for chemiosmosis? Option A, membrane, proton pump, proton gradient, and NAPD synthase. Option B, proton pump, electron gradient, and ATP synthase. Option C, proton pump, electron gradient, and NADP synthase. Option D, membrane, proton pump, proton gradient, ATP synthase. So the correct answer is D. We need a membrane, proton pump, proton gradient, and ATP synthase in energy generation. So option D is the correct answer. Question 7. Melanoate inhibits the growth of pathogenic bacteria by inhibiting the activity of which of these following enzymes? Option A, amylase. Option B, lipase. Option D, dinitrogenase. Option D, succinic dehydrogenase. So, melanoate is an inhibitor, is a competitive inhibitor. It inhibits the growth of pathogenic bacteria by inhibiting which enzyme? Melanoate inhibits the enzyme succinic dehydrogenase. And succinic dehydrogenase is one of the enzymes that is involved in Krebs cycle, TCA cycle. And TCA cycle is known to generate energy. And once these melanoates inhibit energy generation, it will definitely inhibit the growth of pathogenic bacteria. So the correct answer is D, succinic dehydrogenase. It is not amylase. Amylase is involved in enzyme that is involved in starch, breaking down of starch, lipase, breakdown of fat, and dinitrogenase is involved in nitrogen fixation pathway. So the correct answer is succinic dehydrogenase, option D. Question 8. The number of times decarboxylation of isocitrate occurs during single TCA cycle is the number of times decarboxylation of isocitrate occurs during single TCA cycle is 1. Option A, 4. Option B, 1. Option C, 2. And option D, 3. So the number of times removal of CO2 or removal of isocitrate occurs during the single Krebs cycle is 1. Option B is the correct answer. Question 9. What is the net gain when each molecule of glucose is converted to 2 molecules of pyruvic acid? So what is the net gain of ATP when each molecule of glucose is converted to 2 molecules of pyruvic acid? The net gain of ATP at the end of this reaction is 2. So the net gain of ATP when each molecule of glucose is converted, so this is talking about the glycolytic pathway. So the net gain of ATP when each molecule of glucose is converted to 2 molecules of pyruvate is 2. Option C is the correct answer. Question 10. What amount of energy is released from glucose during lactic acid fermentation? During lactic acid fermentation, it is less than 7% of energy that is released. So option A, approximately 15%, no. Option B, more than 18%, no. Option C, about 10%. Option D, less than 7%. So the amount of energy released from glucose during lactic acid fermentation is less than 7%. Option D is the correct answer. Question 11. The number of substrate level phosphorylation in 1 ton of citric acid cycle is option A, 0, option B, 2, option C, 1, option D, 3. So let's look at the question. The number of substrate level phosphorylation in just 1 ton of citric acid cycle or Krebs cycle is 1. Option C is the correct answer. Question 12. Conversion of glucose to glucose phosphate. The first irreversible reaction of glycolysis is catalyzed by. They are asking this question. They are asking of which of the enzymes, all these listed A to D are enzymes. So which of these enzymes is responsible for the conversion of glucose to glucose phosphate? And this is the first step in glycolytic pathway. That's the conversion of glucose, breakdown of glucose through series of reactions in the presence of enzyme catalyzing the reaction. So to pyruvate, that's pyruvic acid. So the first reaction, irreversible reaction, is the conversion of glucose to glucose phosphate in the presence of exokinase enzyme or glucokinase in the liver. So option A, amylase, option B, phosphoglucokinase, option C, aldolase, option D, exokinase. The correct answer is option D, exokinase. Question type 13. The respiratory quotient value of tripalmitin is what? So if you might have come across palmitic acid, so that's responsible in fat pathway. So the respiratory quotient value of tripalmitin. So what is tripalmitin? Tripalmitin is fat, is also known as triacylglycerol or glyceride. So is a storage form of fat in animal. So the respiratory quotient value of tripalmitin is option A, 0.07, option B, 0.09, option C, 0.9, Option D, 0.7. The respiratory quotient value of trypanotene or fat is 0.7. Option D. Question 14. What is the role of NAD in cellular respiration? So NAD is nicotinamide adenine dinucleotide. So its role in cellular respiration, let's take a look at the option. Option A, it functions as an enzyme. No, it functions as an electron carrier. Option B. Option C, it is the final electron acceptor for anaerobic respiration. Option D, it is a nucleotide source for ATP synthesis. So NAD plus is involved, its function as an electron carrier. So that makes option B the correct answer. The correct answer to this question is NAD plus in cellular respiration. The role is it functions as an electron carrier. Option B. Question 15. Oxidative phosphorylation is a formation of ATP by the transfer of a phosphate group from a substance. Option B, oxidation of ATP in phosphate. Option C, addition of a group to ATP. Option D, formation of ATP by the energy released from electrons during substrate oxidation. So oxidative phosphorylation is involved in ATP formation. So at the end, ATP is generated. So formation of ATP by the energy released from electrons removed during substrate oxidation is the correct answer. Option D. Question 16. Which of the following biomolecules is common to respiration-mediated breakdown of fats, carbohydrates, and protein? If you remember during the course of the question, I made mention of acetylcholine. So acetylcholine is a biomolecule that is common to respiration-mediated breakdown of fat. Like the pathway for fat, acetylcholine is generated in protein amino acids breakdown. Acetylcholine is also generated, and this acetylcholine is now used as a starting material in respiration pathway, for example, Krebs cycle and electron transport chain. So the correct answer is option D, acetylcholine. Question 17. Cytochromes are found in A. Crystal of mitochondria, B. Lysosomes, C. Matrix of mitochondria, D. Outer wall of mitochondria. Cytochromes are components of one of the components in the electron transport chain, and that takes place in the inner mitochondrial membrane. So the correct answer is crystal of mitochondria, option A. Question 18. In which of the following processes CO2 is not released? Option A, aerobic respiration in plants. Option B, aerobic respiration in animals. Option C, alcoholic of fermentation. Option D, lactic of fermentation. The correct answer is lactic of fermentation. During this fermentation process, CO2 is not released. Option D is the correct answer. Question 19. In mitochondria, protons accumulate in the A. Outer membrane, option B. Inner membrane, option C. Intermembrane space, and option D. In the matrix. So in mitochondria, proton accumulates pumped into the intermembrane space. So the correct answer is option C. The mitochondrial proton accumulates in the intermembrane space. Option C is the correct answer. Question 20. The energy releasing metabolic process in which substrate is oxidized without an external electron acceptor is called option A, glycolysis, option B, fermentation, option C, aerobic respiration, and option D, photorespiration. The correct answer is fermentation. In fermentation, um, there's no need for an external electron acceptor, for example, the oxygen. So, the energy releasing metabolic process in which substrate is oxidized without an external electron acceptor is fermentation. Option B is the correct answer. Question 21. The aerobic respiratory pathway is approximately termed option A, parabolic, option B, amphibolic, option C, anabolic, and option D, metabolic. So the aerobic respiratory pathway is termed amphibolic. That is a dual metabolic pathway where we have the anabolic pathway and the catabolic. The catabolic is the breakdown and the anabolic is the building up. That's synthesis. So the correct answer is B, amphibolic pathway. Question 22. The overall goal of glycolysis, the Krebs cycle, and the electron transport system is the formation of, when we look at glycolysis, breakdown of glucose, at the end is to give energy to the cell for cell function. Then the Krebs cycle also, the processes is the end process is to generate ATP energy. Likewise, electron transport chain. So the correct answer, let's take a look at the options. Option A, ATP in one large oxidation reaction. B, sugars. C, nucleic acid. D, ATP in small stepwise units. So the correct answer is ATP in small stepwise units. Option D is the correct answer. Question 23. How many ATP molecules could maximally be generated from one molecule of glucose if the complete oxidation of 1 mole of glucose to CO2 and H2O yield 686 kcal, and the useful chemical energy available in the high energy phosphate bond of 1 mole of ATP is 12 kcal. So the summary of this question is to calculate the number of ATP that will be generated from 1 molecule of oxygen if there's a complete oxidation of 1 molecule to carbon dioxide and water. And if that uses 86 kcal, and the useful chemical energy available in the high energy phosphate bond of a mole of ATP is 12 kcal. So how many ATPs could be generated? So what you just need to do is to divide 86 kcal divided by 12 kcal, and then you will get 57 points something, 16. So rounding it up, you get 57. So the correct answer is, let's look at the option. Option A, 1, no. Option B, 2. Option C, 30. Option D, 57. So the correct answer is 57. Option D. Question 24. During which stage in the complete oxidation of glucose are the greatest number of ATP molecules formed from ADP? So during which stage? A, glycolysis, breakdown of sugar, yields about 2 ATP. B, Krebs cycle, also yields about 2 ATP. C, conversion of pyruvic acid to CoA, coenzyme A, there's no energy is generated. Let's say 0. Option D, electron transport chain. that yields about, let's say, 34 ATP. So the question is now asking, during which stage in the complete oxidation of glucose are the greatest number of ATP molecule formed? That is electron transport chain. Option D is the correct answer. Question 25. How many ATP molecules are produced by the aerobic oxidation of one molecule of glucose? So it's just like a follow-up question of question 24. So in glycolysis, two molecules of ATP are generated. Krebs cycle, two molecules, and electron transport chain, about 34 molecules. Summing these ATP that are generated together, that will give us 34 plus 2, 36 plus 2, 38. So the correct answer, option A, 2, option B, 4, option C, 34, and option D, 38. The correct answer is 38, option D. Question 26. Organisms that obtain energy by the oxidation of reduced inorganic compounds are called A. Photoautotrophs. They make their food in the presence of sunlight. B. Chemoautotrophs. Autotrophs, they are organisms that make their food by through inorganic compounds. Option C, saprozoic. Saprozoic feed on organic matter. Then dead organic matter. Then D, coproheterotrophs. They are organisms that feed on fecal matter. So the correct answer is chemoautotrophs. So these organisms make their food by obtaining energy from the oxidation of reduced inorganic compound. So the correct answer is chemoautotrophs, option B. Question 27. Cytochrome is option A, metalloflavoprotein, B, iron-containing porphyrin pigment, D, C, glycoprotein, and D, lipid. Cytochrome is a component in electron transport chain, and it's involved in that's HEME, and this heme contain iron. They are iron-containing porphyrin pigments. So the correct answer is option B, iron-containing porphyrin pigments. Option B is the correct answer. Question 28. In glycolysis, during oxidation, electrons are removed by ATP, option A, B, NAD plus, option B, C, glyceraldehyde 3-phosphate, option C, and D, molecular oxygen. Nicotinamide adenine dinucleotide is involved in glycolysis, and electrons are removed by nicotinamide NAD plus in glycolysis during oxidation. So the correct answer is NAD plus, option B. Question 29. In which of the following do the two names refer to one and same thing? Option A, Krebs cycle and the Calvin cycle. Option B, tricarboxylic acid cycle and citric acid cycle. Option C, citric acid cycle and Calvin cycle. Option D, tricarboxylic acid cycle and urea cycle. So what this question is asking, out of these options, there is one particular option that the two names actually mean the same thing. So the correct answer is tricarboxylic acid cycle, TCA. You will have heard me mention it during the course of the lecture. TCA, tricarboxylic acid cycle, and citric acid cycle. They mean the same thing. Another name for this pathway is also known as Krebs cycle. It is not Kleb, but Krebs cycle. So option B is the correct answer. Tricarboxylic acid cycle and citric acid cycle. They mean the same thing. Question 30. In alcohol fermentation, option A, triose phosphate is the electron donor, while acetaldehyde is the electron acceptor. B, triose phosphate is the electron donor, while pyruvic acid is the electron acceptor. Option C, there is no electron. Option D, oxygen is the electron acceptor. So when we look at alcohol fermentation, triose phosphate is the electron donor, while acetaldehyde accepts electron, is the electron acceptor. So option A is the correct answer to the question. So in alcohol fermentation, triose phosphate is the electron donor, while acetaldehyde is the electron acceptor.
Hi, I'm sure you enjoyed the content you just watched. To see more episodes as that, preparing you for your oncoming exams, smash the subscribe button on this channel right now and visit the playlist that we've made for you, covering over 50 type of different exams, including JAMB, WAEC, Cacao, NEET, IALT, and the likes like that. Also, in every video you watch, we have the link to our apps where you can download on Google Play Store and Apple Play Store and practice hundreds of past questions for free.